So I'm very excited to introduce our next speaker, Dr. Vijay Ramani. Vijay completed his bachelor's in chemical and biological engineering at Princeton University, and after a brief stint in industry, moved to Seattle to the University of Washington to do his PhD with Jay Schenger. There, Vijay conducted numerous innovative studies designing new approaches to RNA proximity, chromatin structure, single-cell indexing, and phenotyping. And then now as a PI at UCSF in the Sandler Faculty Fellow Program, Jay continues to develop new approaches to understand chromatin and gene regulation. And today I'm excited, as I'm sure we all are, to hear about his recent work on massively multiplex single molecule oligonucleosome profiling. So join me in giving a very warm, if socially distant, welcome to Vijay.
Thanks, Ben, for that introduction, and really thanks to the Fragile Nucleosome organizers for giving me the chance to talk to you all today. I think this is such a spectacular community, and even participating in the journal club last week, it's really exciting to see the global chromatin community coming together and sharing their science and sharing their expertise. It really is heartwarming to see that. Okay, so like many of you or all of you here in the seminar, my lab is broadly interested in the organelle that is, I think, beautifully illustrated here. And this, of course, is the eukaryotic nucleus. But more specifically, as Ben alluded to, my interests lie in methods development. And our lab, one of the primary focuses of my lab is to develop sequencing methods to study chromatin structure at high resolution. These include methods for studying 3D chromatin architecture and methods for, as I'll talk about today, resolving the primary structure of chromatin at the resolution of nucleosomes. We all also are interested in chromatin biology, and we're specifically interested in topics related to the linkage between cellular metabolism, transcription, and chromatin structure. And so we're thinking about this in a few different ways, and hopefully in subsequent talks, we might be able to tell you about some of the results of some of these projects. But we're in an ongoing collaboration with Bob Eisenman's group at the Fred Hutch. We're really interested in deeply studying transcription factors that we know are primary integrators of cellular metabolism, and these are transcription factors in the extended MYC network of basic helix, loop helix, leucine zipper transcription factors. We're also interested in understanding how cellular metabolism might directly rewire 3D genome architecture through RNA polymerase 3. And then we're also just broadly interested in the shared usage of intermediary metabolites by chromatin-specific processes and basic biosynthetic processes. These are metabolites like SAM, alpha-ketoglutarate, acetyl-CoA. But as I mentioned before, what really unites all of these topics that we're studying in my lab together is our commitment to using novel molecular methods and developing novel molecular methods to address these questions in a way that perhaps was not addressable before. But today I am really going to focus on a method that we're quite excited about. This is a method that we've posted up on bioRxiv as of a couple of months ago. And the motivating factor for developing this novel method really is the question of how nucleosomes are arranged in the context of chromatin on single molecules. As you all know, the primary structure of chromatin, that is the wrapping of DNA about octameric nucleosome complexes, is essential to the templating of nuclear processes that are important for life itself. Like the relative accessibility of DNA to various trans factors important for cell type specific transcriptional regulation, but also processes like DNA replication and DNA repair is determined in part by the relative arrangement of nucleosomes and the compaction of nucleosomes in various contexts. And so there's a rich methodological history in terms of studying the primary structure of chromatin and studying nucleosome patterning in chromatin. Many of these methods involve a favorite enzyme of mine, the endo-exo micrococcal nuclease from Staph aureus, And what one can do via nucleases like micrococcal nuclease is take nuclei, digest nuclei with a range of concentrations of this endoexonuclease, and result in a ladder of digested nucleosomal fragments that can then be analyzed via gel electrophoresis or southern blotting to look at the relative periodicity and regularity of chromatin fibres. These are, I think, decades of work have suggested and shown just how valuable these approaches are, but they do have some limitations that are worth bringing out. First, these methods do inherently destroy the chromatin fiber in order to make the measurement. One is digesting chromatin with a nuclease, often to quite small repetitive subunits. But also, and I think this is another important point to highlight, that these are really bulk measurements. You're taking many nuclei, digesting them with your enzyme of choice, and these analytical gels or southerns, right, are representative of an average of the molecular state or the nucleosome positioning states of many fibers averaged together. In the last decade, these approaches, which as I said, were typically read out using gels or first, if one were interested in probing specific regions, gels plus southern blotting, have been converted to genome-wide assays using Illumina sequencing as a readout. But these sequencing assays still suffer from the same drawbacks. We're often, whether we're looking at MNase-seq data, DNA-seq data, or even Daria alluded, I think, quite nicely to the power of now attack-seq data for potentially measuring both chromatin accessibility and open chromatin and nucleosome positioning. You're still fragmenting chromatin and analyzing usually mononucleosome and smaller sized fragments. And so this is akin to taking the image that or the painting I showed you before and really chunking it down into smaller mononucleosome sized fragments, when really we'd like to move towards developing a more complete understanding of how chromatin is arranged on single fibers. And so there are, in fact, approaches that allow for single molecule probing of chromatin structure. These are approaches that were really pioneered by the Jones, Clada, and Schubler lab, among others. And these are approaches that use methylation to actually mark the presence and absence of nucleosomes on single chromatin fibers. And so in these cases, these are approaches like GNOME-seq or DSMF, where GPC or CpG methyltransferases are used to methylate cytidines in various nucleotide contexts, and then bisulfite sequencing is then used to mark where those modifications occur. These this is another powerful set of assays, but these also share some biases. One has to use GPC or CpG methyltransferases to induce these marks. It becomes problematic when there is endogenous CpG methylation in the organism of interest. And also one has to use bisulfite conversion, which is inherently quite harsh and can lead to shorter fragments. This is without even mentioning the fact that the Illumina sequencer actually has its own biases and can only sequence, thanks to the bridge PCR chemistry that is used for Illumina sequencing, can only really allow for sequencing of fragments that are at an absolute maximum of, let's say, 700-ish nucleotides, right? And so what we'd really like is to leverage some of the really nice methods development work that's been done for footprinting chromatin, but pair it with a platform that allows for the sequencing of longer molecules than one would be able to sequence on an Illumina sequencer. And so luckily there is a platform available to do this, and that's a single molecule real-time sequencer from Pacific Biosciences, which allows us to resolve long, and by long I mean anywhere from 10 to 20 kilobase long molecules at very high accuracy. And so next like couple minutes, I'll just very quickly go over how this platform works, because I think it's very cool and it's quite important for the technique that I'll talk about. And so PacBio sequencers are comprised of the sequencing platform itself is an array of zeptoliter scale wells that enable the detection of single molecules worth of fluorescence. One embeds or immobilizes covalently closed circular DNA bound to engineered rolling circle polymerase in these zeptoliter scale wells, such that there's one molecule per well, and then a polymerase reaction is allowed to continue in the presence of fluorophore conjugated nucleotides. And so, as the polymerase processes and incorporates these nucleotides, the fluorescence of these individual nucleotides can be read out, and a movie can be taken. And as these, and so the sequencing is carried out as these pulses are read out, one color per nucleotide. And so this happens in an array of millions of zero-mode waveguides. And so now, these wells are called zero-mode waveguides, or ZMWs, and this chemistry allows for the highly accurate sequencing of single molecules, of single long molecules in real time, hence the name single molecule real-time sequencing. A really interesting wrinkle of this technology is that the kinetics of the polymerase moving across these templates can actually be measured by the sequencer during the course of sequencing, and are actually perturbed by DNA modifications like adenine methylation. And that can be seen in this figure here, where templates were used harboring unmodified or modified adenine residues. And in the presence of in this case, M6DA, a pause is actually induced over the course of sequencing by the modification. And so, very simply, we asked, could we actually combine smart sequencing with methylation footprinting to non-destructively profile nucleosome positions on single chromatin fibers? And so this is work that was really spearheaded by a graduate student in my lab, a computational postdoc in my lab, and this was done in close collaboration with Geeta Narlakar's lab upstairs, in close collaboration with a postdoc in Geeta's lab, Dr. Laura Shieb. And so we first wanted to see if this could work at all, and so it made sense to start with a really well-defined template. So with Laura and Geeta's help, we assembled chromatin onto a 9-mer array of 9 tandem repetitive, seated on 9 tandem repetitive copies of the Wicom 601 nucleosome positioning sequence with a known linker length of 46 nucleotides. We then exposed those arrays to a non-specific adenine methyltransferase called EchoG2. And the hope was that we would see changes in a chromatin-specific way in one of the 2 kinetic parameters that the PacBio sequencer reads out. So the PacBio sequencer can read out these 2 parameters. One is the pulse width, which is the amount of time that the nucleotide is sitting in the active site of the polymerase, and then also this interpulse duration, which is this pause as the polymerase is transiting between nucleotides. It turns out that pulse width doesn't really matter, but the IPD is super instructive for detecting these modifications. And so just to bring this home, what we're doing, we have DNA that at one point was chromatin. Hopefully we're methylating the accessible linker sequence in between known well-positioned nucleosomes on single fibers. The polymerase transits, it pauses at these adenine, at these methylated adenines, and hopefully we should see a change in this interpulse duration that I mentioned that is consistent with where nucleosomes were. So turns out this works. I'll move very quickly through this kind of proof of concept data, but we see really beautiful changes in IPD that are chromatin specific. We also have controls where we have naked DNA that isn't methylated and then naked DNA that is completely methylated. And we nicely see that chromatin samples fall in between those. It's also chromatin specific and adenine specific. So I'll just highlight here where we can see that adenines that fall outside of the Wicom 601 positioning sequence happen to be methylated, and we see a change in the IPD, but this is not the case in adenines that should be protected by nucleosomes. We can come up with pretty straightforward analytical pipelines for calling expected dyad positions, and we see that those fall quite close to zero. The statistics are in our preprint. And we can also nicely call nucleosome repeat lengths. We expect a nucleosome repeat length in this case of 193 bases, and that's precisely what we see, both if we take single molecules and average the 9 or 8 nucleosome repeat lengths between nucleosomes, but also in terms of single dyad to dyad distances on these templates. And so this seems to be working quite well. We're really excited by this. And another, I think, really cool aspect of this technology is that I mentioned that these are covalently closed circular templates that are immobilized on these zero-mode waveguides. And so what the polymerase is doing is it's going around and around in circles. And so you actually read single molecules many times over the course of sequencing. And so we can actually detect these changes in these kinetic changes many times and use that to increase our modification detection fidelity. And so we're quite pleased with the accuracy that we're getting in terms of really being certain that the residue that we're sequencing is truly modified. And so this is again in close collaboration with Geeta's lab, and we're very excited about using just this in vitro platform as a new platform for studying a single molecule chromatin biochemistry. And so we can do experiments where we say take a panel of molecules, of chromatin molecules, sequence them, and here I've sectored out about 3,200 molecules that in each row here is a single molecule now. And this is unremodeled chromatin, and we can expose it to an ATP-dependent chromatin remodeler and actually look at chromatin remodeling outcomes on individual chromatin fibers with single molecule resolution. So we're incredibly excited about the possibilities associated with this approach. And so this seems to work. We needed to come up with a name for this. We were unsatisfied with many names. We ended up coming up with a favorite food of mine, the SAMOSA, and that's short for single molecule adenine methylated oligonucleosome sequencing assay. And so in the next 8 minutes or so, I'm going to walk through our early attempts to apply SAMOSA. This was all in vitro, but now we'd like to apply SAMOSA to study a chromatin structure in vivo. And so we know this works on chromatin fibers. The next question is, how do we get intact chromatin fibers from living cells? And so we were inspired by work from the late 90s and early 2000s by Nick Gilbert and Wendy Bickmore, where they found that they could incorporate a very light MNase digest, dissolve the nuclear membrane through fusogenic lipid treatment, and then simply solubilize via dialysis long intact chromatin fibers into solution. And so we just basically borrow this protocol. We take nuclei, we perform a limited MNase digest. We then take these MNase-derived oligonucleosomes, methylate them with Echo G2, as we had done with our in vitro-derived fibers. And then now we actually have some more information here compared to our in vitro experiments. We actually have MNase-protected ends that should mark barrier elements across the epigenome. We should mostly be nucleosome-bound elements. And then along single molecules, we have this M60A signature that should footprint nucleosomes themselves. And so we think this is kind of cool because it is this kind of dual omic measurement of these barriers and then single molecule nucleosome DNA interaction. And so we can look at these data in two ways. The MNase data, I'm going to color in purple. The methylation data, I'm going to color and green for the rest of this talk. And so if we just look at the fragment length distribution of our molecules, we see that they really beautifully correspond to integral multiples of successively longer oligonucleosomes. This data actually, I think, is highly comparable to really nice work from Peter Becker's lab on the array-seq approach that uses nanopore to sequence MNase-derived oligonucleosomes. They also see a similar ladder, and so we think that this data can be used for many of the same analyses that they used. But also, when we simply look at the average methylation pattern along these molecules, say out to 1 kb, we also really nicely see nucleosomal periodicity in the methylation channel. And this, of course, we don't see if we look at controls where we've either not methylated the DNA or we've taken naked DNA and methylated it. And all of these experiments, I should note, have been done in the really well-characterized K562 human cell line. Okay, so for the next 5 minutes, I'm going to briefly talk about some pilot experiments we did in our preprint to demonstrate some interesting things that we see in our in vivo SAMOSA data. This is all in greater detail in the preprint, and I'm happy to answer questions in the Q&A section about each of these sections. So first, we were interested in just seeing what is the variability and variance in nucleosome spacing and regularity along these individual chromatin fibers genome-wide. And so to carry out this analysis, we borrowed concepts from single-cell analysis, and actually just took each of our individual molecules and clustered them using a community detection algorithm that is typically used for single-cell analysis. And what we found was that these clusters, which these 7 clusters are illustrated here, nicely broke up the epigenome across both nucleosome regularity and nucleosome spacing. So we have clusters, like clusters 1 through 3, which I can also show here, that are fairly irregular. That is, in our inferred nucleosome repeat lengths, there's a fair amount of variance. And we also had a set of 4 clusters that nicely stratified by increasing average nucleosome repeat lengths, which we can kind of see this distribution across single molecules here. We can also visualize these as single molecules, and we can visualize these violin plots as distributions. And I've just selected 3 clusters here just to give you all a sense of what these data actually look like at single molecule resolution. We also did pilot analyses looking at variation in nucleosome spacing at predicted transcription factor binding sites based off of ENCODE data. Our data are, I think, this is another interesting case where having the MNase data and the M60A data is somewhat interesting. We can look at the data in a couple of different channels. We have MNase cuts that are highly reminiscent of MNase-seq data in terms of the patterning of ends with respect to predicted transcription factor binding sites like those for CTCF or NRF1. But then we can look at individual molecules that happen to harbor transcription factor binding sites and examine changes in methyl 6-DA signal at single molecule resolution. Here I'm just looking at the averages. We can compare these to controls where we can make sure that these are not simply, the signals we're seeing are not simply based off of, say, the dinucleotide content of the molecules we're looking at. But because we have single molecule information, we can, again, cluster all of these individual molecules and break these down into various nucleosome-occupied states flanking individual predicted transcription factor binding sites. And so we speculate on what this might mean in our preprint, but we do think we're seeing both a stratification of highly methylation-resistant sites, various types of nucleosome-occupied sites, which we think are different registers about about transcription factor binding sites, and then sort of focally accessible sites with nucleosomes flanking the transcription factor binding motif, and then what we think might be just hyper-accessible sites where there's a massive amount of accessibility on that template to the methyltransferase. we can again look at these and see that these results are truly robust at sort of single molecule resolution. I'm just sampling 500 molecules for a few of these clusters here. And we think that perhaps this could represent transition states between sort of nucleosome occupied and perhaps stochastically accessible templates, more focally accessible templates with nucleosomes flanking the transcription factor binding motifs, and then perhaps these might transit in sort of a reversible way between more hyper accessible sites that could allow for more transcription factor binding. And then we can also look at this across human epigenomic domains. I'm going to go very quickly through this, but we can take the clusters that we identified earlier. We can examine how these clusters break up across, say, constitutive heterochromatin, facultative heterochromatin, active enhancers and promoters, and then gene bodies that are actively elongating, right? I think the takeaway here that we're really interested in hearing more about and discussing, right, is that all of these epigenomic domains are comprised of a highly heterogeneous mixture of chromatin fiber types, stratifying many different nucleosome repeat lengths, as well as intrinsic regularity. If we look at these domains, we do in fact see that there's enrichment and depletion across these epigenomic domains, but the effect sizes are quite small. And so we are really interested in looking at this further and really thinking about how really heterogeneous chromatin fiber types might give rise to the epigenomic domains and active and silent states that ultimately encode the cellular states that we know that they're associated with. So with that, I'll wrap up. So I've talked to you about SAMOSA, which allows for non-destructive detection of nucleosome DNA interactions on individual molecules. We've done some proof of concept in vivo experiments to provide in vivo estimates of nucleosome regularity and spacing on single molecules genome-wide. We think this is a really nice complement to the multitudes of epigenomic data that have been generated via Illumina. We're interested by this finding that epigenomic domains are composed of a pretty heterogeneous mixture of fiber types. And I think I'm very excited by the notion that third-generation sequencing can now be used to make really high-throughput epigenomic measurements. I want to call out that there's really beautiful work from Will Greenleaf's lab, Kin Fai's lab, Winston Tim's lab, John Stam's lab most recently, that all have very similar approaches that use either nanopore or PacBio to make these measurements. And so I really think this is going to be a fruitful way forward for epigenomic measurement. So with that, thank the lab. Again, Noor, Colin, and Laura really spearheaded this work. And with that, happy to take questions. Thanks for your time. And sorry I went a couple minutes over.
Awesome. Thank you so much, Vijay. That was awesome. I have so many questions, but we have a few to begin with from the audience. And so I'll remind everyone that you can either type your questions into the Q&A box, or if you raise your hand, we'll unmute you and you can ask your question yourself. So Irene Kaplow asks, are different fiber types associated with binding of different transcription factor families?
Yeah, so that's a great question. So they are. We don't have enough coverage at the moment. So as we sequence more, I think we'll be able to answer that question in a way that is at least satisfying to me. In our preprint, we show that the relative enrichment of different fiber types and the relative enrichment of different nucleosome-occupied states about motifs differs significantly across the different transcription factors that we tested, right? But I think in terms of correlating those differences with like a satisfying biochemical parameter, like the residence time of these transcription factors in vivo, will require greater sequencing depth. So I don't want to say anything concrete about that quite yet.
Yeah, and at this point, can you comment on for a given region, the variation molecule to molecule, or is that the same sequencing depth limitation?
So I think for epigenomic domains, because we can average across many molecules across very large domains, we can say something, right? And I am fairly confident in this finding that the domains are highly heterogeneous, right? Mm-hmm. And where I think our findings coincide with the the large amount of epigenomic data that shows that NRLs are different by just MNase-seq across these domains, right? Is that when we look at enrichment of usage of these fiber types, we see significant enrichment or depletion, right? But the effect sizes are I think, interesting to think about in terms of their size. The difference between constitutive heterochromatin and active chromatin in terms of the usage of, say, a fiber with an NRL of 195 nucleotides, for example, is only, say, 4%.
Yeah, it's interesting. Okay, we've got a question from Jason Fan, who's raised his hand. So I'm going to allow Jason to talk, and he can ask his question.
Hi, can you hear me?
Yeah.
Oh, yeah. Very cool technique. Really exciting. I was curious at say at highly transcribed genes, can you see with your sequencing like maybe like an increase in overall accessibility of the gene body, or perhaps can you capture like a Pol2 footprint with this?
I hope so, right? I think we need. So we need to sequence deeper, right? And these are experiments we're doing in the lab now. It's worth mentioning that John Stam's paper has sequenced S2 cells quite deeply, right? Their paper doesn't do any type of Pol2 analysis, but it would be interesting to look at their data and maybe repeat some of the analyses that Arno Krebs did in his SMF paper from Dirk Schubler's lab on that. It's something we're certainly thinking about. We'd like to do. What we can say right now is that if we look at H3K4 trimethylated regions, right, we see the characteristic hallmarks of disorder that one would expect for NDRs, right? But beyond that, I think we need to sequence deeper to really correlate, how to first see if we can see Pol2 footprints, right, and then kind of make conclusions based off of that.
Great, thank you.
All right, and I've got a question here from Srinivas Ramachandran. Apologize if I butchered your name. Could the presence of all clusters and all epigenomic domains be telling us about the kinetics of nucleosome positioning genome-wide?
So the SNAP interpretation, right, is that it's a highly heterogeneous landscape, right? That in fact nucleosomes, even in heterochromatin, right, there's a fair amount of heterogeneity and a fair amount of dynamics present or associated with, at least in the context of heterochromatin, what I think has been largely thought of as a reasonably static entity up until fairly recently, right? And so I think thinking about or contextualizing these results in the context of maybe some of the dynamics that labs like Geetha's lab have begun thinking about and is something we're very interested in doing, right? But for right now, I would like to think that this is suggestive of a highly dynamic nucleosome DNA interface, even between heterochromatin and euchromatin.
Awesome. Okay, and we've got a question from Katie Galloway asking, are other DNA modifications detectable beyond methylation?
Yeah, so that's a great question. 5-methylcytidine is not robustly detected, which is great in our case, right? Because we get to use a modification that isn't abundant at all in mammalian epigenomes, right? And is highly detected, right? I think some mutagenic modifications are robustly detected, but at the cost of quite literally stalling the polymerase. And so you have this trade-off of like not really being able to effectively sequence your template. But 5-methylcytidine is not. If that's where the question was going, I guess I don't know.
Sounds good. Sounds like a good answer to me. I'm not sure. That's interesting. So we've got a question from Arpit Mishra, who asked, have you looked at G-quad sites?
I have not, and would be interested to offline hear more about why one would be interested in G-quads and what specifically you might be interested in, but we haven't thought much about them at the moment.
And then he also asked how this method correlates with FibreSeq.
Yeah, so the FibreSeq paper came out like 3 weeks ago or something, right? So we are in the process of downloading the data and comparing to it. So I don't know yet is the answer.
Yeah, and then we've got a question from Ben Weekly who says, amazing talk, and asks about how much input you need from cells in order to perform SAMOSA, and is this something that you can do at the single cell level?
So input-wise, we can make PacBio libraries from 150 to 300 nanograms of material, right? So I'm confident that we could get down to that level, right, in terms of input. We've been using K562s because they're easy to grow, but we'd like to apply these to maybe cases where things are more input limited. Single cell is a challenge though, because PacBio libraries and this method, in fact, is hinges on not doing PCR, right? We're natively detecting the modification on the parent template, and so one would have to come up with a way to do single cell without doing PCR, which is quite challenging. But if one had a way to do it, I'd be all ears. Yeah.
Okay, awesome. So I think that's all the time we have for today. Thank you to both our speakers for wonderful talks, and thank you all for tuning in.